Hello, everyone. Let's go ahead and finish up chapter 11. So we'll start off just with some basic points here about disuse atrophy. So you remember what atrophy means. All right, atrophy means that you're having a decrease in cell size, and eventually those cells will undergo apoptosis, leading to an overall decrease in tissue mass. Okay, so if you're not using your muscle cells, this is what's going to happen. If no stimulation is given to a muscle cell, then there's no point, there's no purpose for that muscle cell any longer because it functions strictly on stimulation or excitation from a neuron. I mean, at least when you're dealing with skeletal muscle. Okay, so if you're not using them, you will lose muscle mass. Now, what are some causes to this? Well, immobilization, typically due to some sort of damage to the spinal cord, for example, or to nerves, for example. All right, if you sever a nerve that is supplying a muscle, then you're not giving any stimulation to that muscle, so the muscle will atrophy. All right. So, just this bottom point down here, this is why rehabilitation, if someone has an injury like that, maybe they've had some spinal cord damage, it is really critical that they undergo physical therapy as soon as possible and as often as possible. Okay, the sooner they do that and the more frequently that they do that, the more likely that they are not going to experience, or they will experience to a lesser degree, this atrophy or loss of muscle mass, right? And of course, if scar tissue develops, if we have fibrous connective tissue replacing the damaged muscle, then we cannot do rehabilitation. All right, if there's scar tissue there instead of muscle tissue, there's nothing there to stimulate. This slide, I'm not going to ask any questions about this slide. It's a good thing because it's kind of blurry. But, I really just put this in here for your interest. If you have questions about this or curiosities about, some of the details about muscle fatigue, all that I ask is that you just know basically muscle fatigue is referring to weakness of the muscle due to prolonged use. And again, if you're interested, here are more details about that, but I will not ask any questions about it. Muscular strength. What does strength of a muscle depend on? Well, temporal summation. Now, it's not listed here on this slide, but I did discuss it earlier. Spatial summation also is going to be a big predictor of muscle strength. Okay, so don't forget, not just temporal summation, okay, but spatial summation as well will determine the strength of a muscle. The length tension relationship, all right, so that's also going to help determine muscle strength. Okay, how much tension is generated and how that relates to how the muscle is going to change in length. And then fatigue, of course, will play a role in strength of a muscle. All right, so moving on, we're going to talk a little bit about smooth muscle. All right, we will talk about how smooth muscle contracts. So you will have a couple of questions on the details of that. You understand skeletal muscle contraction, which is much more complicated. So I feel like smooth muscle, you should get this pretty well. So for your exam and quiz, pay attention to what's different about skeletal muscle versus smooth muscle contraction. All right. Be sure you know the differences between the two, as well as the details of each. All right. So it says here, injured smooth muscle does regenerate well, right? Unlike skeletal muscle and cardiac muscle, smooth muscle has a pretty good regenerative capacity. It does do mitosis um, uh, throughout life and more frequently than skeletal and cardiac. All right, cardiac muscle really is not going to regenerate. Skeletal muscle can regenerate. It's possible, but it's not going to be great, and it's going to take a long time. Okay? So, but smooth muscle is pretty good at regenerating if you do have damage to smooth muscle. Now, smooth muscle contraction compared to skeletal muscle contraction and cardiac muscle contraction is a little bit slower, right? It's not as quick. But at the same time, the contraction will last longer, all right? And we don't experience fatigue in smooth muscle. Remember your location for smooth muscle. All right, hollow organs, blood vessels. Now, let's mention, you'll probably have a question about this. There are going to be lining these tubular structures such as intestines, blood vessels, etc. You will find actually two layers of smooth muscle. You have a longitudinal layer and a circular layer. Okay, the longitudinal layer is what will cause this organ or structure to shorten. The circular layer will cause the lumen to become more narrow. Okay? And now what we're going to see with smooth muscle is that we have an alternation in contraction of these two layers. Okay, so the longitudinal layer will contract, causing the organ to shorten or the structure to shorten. And then right after that, immediately after that, you'll have a circular layer contract, causing the lumen to become more narrow. And then back again to longitudinal layer contraction. So basically, these just sort of alternate, right? Now, this is called peristalsis. It's a special kind of movement or contraction that's going to take place in these hollow organs, like the intestines, for example. All right, you'll have alternating contractions and relaxations of these layers. And what this allows to happen is it allows something to flow through that lumen. Right? It's going to basically, in the example of digestive system organs, it will, this alternating contraction between these two layers is going to help propel food along, right? The esophagus is really good at this, right? We after we swallow our food, the esophagus, the two muscle layers of the esophagus will alternatively contract or alternately contract, and this will help propel the food substances down to the rest of the digestive tract. And so you should know that that's called peristalsis. Just a closer look here. So this is showing the small intestines, portion of that. And the outer layer here, okay, the outer layer, this is your longitudinal layer. Okay, so remember, the longitudinal layer, when it contracts, it's going to cause a shortening of the muscle here along the whole entire organ. You will then see contraction of the circular layer. The circular layer is the inner layer here. And when this contracts, it causes this lumen to constrict or become more narrow. All right, this is just showing you again. Now, you will not be asked in this class to identify these layers histologically. In Bio 139, you will be asked to identify all of these parts. Another blurry slide. Well, let's see what we can get through on this. So, microscopically, how does smooth muscle compare to skeletal muscle? I'm sure you'll have a question about it. Now, the smooth muscle does not have a neuromuscular junction. It does not have sarcomeres. Okay? There is no sarcomere arrangement, nor does it have T-tubules. Now, although we don't have a sarcomere, we do have thick and thin filaments, okay? But they're not arranged into a sarcomere, okay? And again, remember, it's really the sarcomeres that give skeletal muscle and cardiac muscle that striated appearance. And so with smooth muscle, we call it smooth because you don't see any striations. And this is why we don't see these striations, because thick and thin filaments are not arranged into a sarcomere. They're not aligned with each other like that. All right, a certain part of the nervous system called the autonomic nervous system is going to be what innervates smooth muscle. So the autonomic nervous system, all right, will be responsible for controlling smooth muscle. The autonomic nervous system is typically associated with involuntary sorts of actions. You do have sarcoplasmic reticulum in the smooth muscle, but it's not as developed. It's just going to have a different structure. It's not as intricate as you would see in skeletal muscle. Yes, of course, the SR here in smooth muscle has the same function. It is going to store calcium, and you will see calcium release coming from the sarcoplasmic reticulum, like you saw in skeletal muscle. But really a very important point here that you should know, and it's really hard to see down here, but smooth muscle is really unique compared to cardiac and skeletal muscle when it comes to contraction, because contraction is based on this phenomenon known as calcium-induced calcium release. Okay, so remember when we looked at skeletal muscle and we saw that you have sodium influx into the muscle cell, right? Remember, through the ACH receptor that turns into a sodium channel, sodium comes into the muscle, and sodium will participate in changing the voltage on the inside of the cell, and that causes voltage-gated calcium channels on the SR to open so that calcium could come flooding out to the sarcoplasm. You remember that part of skeletal muscle contraction. Well, you're going to have in smooth muscle a similar deal where you have calcium channels, voltage-gated calcium channels on the SR in smooth muscle open, and you'll have calcium come out. Yes, that's true. But instead of sodium being the ion that triggers that process, instead it's going to be calcium in this case. Okay, so with smooth muscle, instead of sodium influx, we're going to be having calcium influx. And again, calcium has 2 positive charges on it. So this will lead to a change in the voltage on the inside of that smooth muscle cell, and then lead to opening of these calcium channels on the SR and efflux of calcium from the SR, and so on. Okay, so big difference here between smooth and skeletal muscle is the fact that we have this very unique situation where we have calcium-induced calcium release, right? Calcium-induced calcium release. So calcium's coming into the smooth muscle cell, and that is causing more calcium to be released out of the SR. Okay, so that's what's meant by calcium-induced calcium release. Okay, so just a little more here about what's different between smooth muscle and skeletal muscle. Okay, so, as mentioned, we do have both thick filaments and thin filaments in smooth muscle. There are going to be fewer thick filaments. Okay, I'm not going to ask that you know the ratios here, just that there are fewer thick filaments compared to skeletal muscle. Now, Z discs are absent in smooth muscle. We do not have Z discs. Why do we not have Z discs in smooth muscle? Because we don't have a sarcomere. So there's no need to have these Z discs. Okay, but in place of that, we do have something called dense bodies. Okay, so dense bodies are going to be these protein structures that are going to help anchor the filaments to the sarcolemma in smooth muscle. Okay, so the dense bodies in smooth muscle are kind of comparable to the Z disc of skeletal muscle in the fact that they both are going to serve to anchor the thick and thin filaments, okay? But again, we don't have a sarcomere, so we don't have these Z discs, instead dense bodies. There is no troponin complex. Okay, very different here from skeletal muscle. We have tropomyosin, okay, but we do not have troponin. So although we're still having a similar deal where we have calcium coming out of the SR, calcium is going to bind to a different protein. Okay, we don't have troponin there. Instead, we're going to have this protein called calmodulin. But calmodulin does not have the same role that troponin had. Okay, so what's different? No troponin. And we have calmodulin instead, which is sort of like the counterpart to troponin in the sense that calcium is going to bind to calmodulin. This is a picture of a smooth muscle. Two smooth muscle cells are shown here. Remember, they're going to be like this spindle shape with oval nuclei. And when it contracts, okay, like other muscle cells, you will see a shortening here. As far as the mechanism of contraction of smooth muscle, like I said, it is going to be slower. So, okay, it's going to be more of a slow sort of contraction, more of like a steady, longer lasting contraction. It will be slower and longer lasting and very synchronized. Now, something else that's different here, we have gap junctions present here in smooth muscle. You also see gap junctions in cardiac muscle, and you'll talk about that next semester. So gap junctions are seen in smooth muscle, and this allows us to have transferring of our ions, mainly calcium, to and from your muscle cells. Now, there is a lot of autorhythmicity that goes on here in smooth muscle because, again, smooth muscle is involuntarily controlled. So they sort of have their own little pacemaker inside that's going to help maintain muscle tone. Again, this is involuntary. Okay. But rate and intensity of contraction is going to be controlled or affected at the end of the day by the autonomic nervous system. Okay, and again, we have these blurry slides, but really the main idea here on this slide is just reiterating that smooth muscle is going to be stimulated and start the contraction process based on calcium influx. Okay, so calcium from the extracellular fluid is going to come rushing into the smooth muscle cell, and this will cause voltage-gated calcium channels on the SR to open, and then we have our calcium coming out. Okay. Now remember, so we'll have calcium channels all along the sarcolemma, allowing this calcium from the extracellular fluid to come in. And, there are different signals to cause this, usually change in voltage. We can have binding of neurotransmitters, for example, to the surface of the muscle cell that can cause this as well. All right. Now, there is still the sliding filament mechanism at work because we are having a shortening of the muscle, similar to what we see in skeletal muscle. All right, but again, we're really a big difference, again, just reiterating it again, calcium-induced calcium release, because this really is sort of a unique phenomenon that you don't see too often in the human body elsewhere. ATP will similarly be used to energize this process. Now, and similar to skeletal muscle, contraction will stop when calcium is no longer available to bind to our protein called calmodulin. Okay. So again, we are still going to have calcium coming from the SR through calcium-induced calcium release. But remember, some of that calcium coming from the extracellular fluid is also going to be used for to start the contraction process. Calcium will bind to this protein calmodulin. And then actually, let me jump ahead to the picture before actually saying these details, because I think that's probably a little easier. I wonder if I can zoom in on this. Okay, yeah, let's zoom in on this a little bit. So, okay, you must know the steps of smooth muscle contraction, just like you did with skeletal muscle. So what are we going to have happen here? First, we're going to have calcium influx. All right, so calcium is going to be entering through the sarcolemma into the sarcoplasm of the muscle cell. All right, so calcium ions come in, and that calcium will be used to bind to this protein here called calmodulin. This calcium that comes into the smooth muscle cell will also be used for calcium-induced calcium release, where because we have these two positive charges on each calcium ion, that's going to change our voltage. That's going to cause the opening of voltage-gated calcium channels on the SR to open, and then we have calcium coming out of the SR. Okay, so we're having calcium coming from the extracellular fluid. We're also having calcium come from the SR. Okay, so plenty of calcium there. And so this calcium then is going to come and bind to this protein called calmodulin. That ends up activating the calmodulin. Once we have our calmodulin activated by calcium, then calmodulin is then going to start a series of activation steps. So activated calmodulin will then activate something called, you must know this, it's called myosin light chain kinase. Okay? It's an enzyme. Okay? It's called myosin light chain kinase. So the calmodulin, once it is activated through the binding of calcium to it, it will then activate this kinase. You remember what a kinase is? A kinase is an enzyme that is going to add a phosphate to something. Okay? So now this activated kinase is going to add a phosphate. Okay? It's going to add a phosphate. Okay? To the myosin head. Okay? So see what's happening here. Okay? The activated kinase is actually going to take a phosphate from ATP, and it's going to transfer that phosphate from ATP over to the myosin head. We can now say that the myosin head is phosphorylated or activated. Okay, so we're phosphorylating the myosin head here. Okay, because the kinase takes a phosphate off of ATP, transfers it over here. We get the conversion then to ADP. Okay, so look that ATP is also needed for this process. Okay, so now once our myosin head is phosphorylated, remember we're adding something to this myosin protein. So this causes the protein, the myosin protein, to change its conformation. So it will cause it to sort of lift up. All right. Remember how when we saw with skeletal muscle that hydrolysis of ATP on the myosin head caused the myosin head to move upwards, to sort of lift up so that it could be close to the thin filament. Well, we have a similar deal here. But instead of hydrolyzing ATP, what we're doing instead is we're using a kinase to take off that phosphate and add that phosphate to the myosin head. All right, and then what we see here is the same thing that we have seen before with skeletal muscle. All right, we're going to have our myosin head reach upwards or downwards and bind to the thin filament and shorten the sarcomere. So let me go back and make sure I said everything. Right. Okay, and I basically said all this, remember. So we're going to have calcium coming into the muscle cell. That calcium, as well as calcium retrieved from the SR, is going to bind to calmodulin. That will activate calmodulin. And then that calmodulin will then activate a kinase called myosin light chain kinase. And this kinase then will remove a phosphate from ATP and add it to the myosin head. all right, which allows this myosin head to lift upwards and be able to form a cross bridge with actin. How do we stop smooth muscle contraction? Well, we need calcium to detach from calmodulin, right? And we're also, similarly to skeletal muscle, we're going to actively transport calcium back into the SR, and we're also going to send some of it back out into the ECF, right? Active transport means requiring energy. Remember that this is a calcium pump on the SR. Calcium pumps require ATP. We're going to do dephosphorylation of the myosin head. So we're going to have a phosphatase come along and remove that phosphate from the myosin head, and that renders the myosin head inactive at that point. Okay, so that's the basic mechanism there of smooth muscle contraction. You should just take note of the differences and similarities between skeletal and smooth muscle contraction. Not too much on this slide. Smooth muscle is involuntary, right? But, so how's it going to contract? It's going to contract in response primarily from, as mentioned, from input from the autonomic nervous system. Okay, the autonomic nervous system. Now, I'm not going to ask you the details here that are listed under here. I'm not going to ask you about that. This is something, because it's really going to take us into a whole, deeper level of the story. And you will be studying this in AP2, so I'll let you do that then. All right. Other things that can activate contraction of smooth muscle. Hormones. Yeah, we have a variety of hormones that can stimulate smooth muscle contraction. And we actually talked about one of these way back in chapter 1 when we talked about homeostasis. We mentioned oxytocin as a hormone that will stimulate contractions in the uterine muscles. Other factors that can stimulate smooth muscle cell contraction would be carbon dioxide, oxygen, different pH levels, temperature, degree of stretch. Okay, I'm not going to ask too many details about this. All right, just have an idea. Yeah, we can have these different factors affect smooth muscle contraction. All right, so muscular dystrophy. First of all, muscular dystrophy is a group of diseases. All right, there are a few different types, but we're just going to mention the most common one, the one that, in the clinical world you would be most likely to see, and that would be Duchenne muscular dystrophy, or DMD. All right, this is an inherited disorder. Inherited. So they are born with this problem. Okay, so you're going to see this because it's inherited. You're going to see the symptoms of this early, okay, very early, usually a couple of years old. You're going to start seeing symptoms of this disease when the child starts to learn how to walk, or right around the age where they're trying to learn how to walk, to stand up, things like that. So what's the problem here? We have a mutation in the gene for dystrophin. Right? So remember what what dystrophin does. Dystrophin is going to link thin filaments to the sarcolemma, right? But if you don't have dystrophin, well, you're not going to have a proper structure associated with the sarcomere and the sarcolemma. Okay? So what ends up happening, because this structure is faulty, the cell membrane is actually going to start to get destroyed whenever we do contraction, or attempt to do contraction. And again, remember, this is happening at 1-2 years old, okay, and the child is attempting to move. So every time they attempt to move, because of this faulty structure, it's actually going to end up destroying their cell membranes of the muscle cells. So because we end up getting damage to these membranes, necrosis will start happening, and scar tissue will be the result of this. Okay, so it's basically, the point is it's a sort of a gradual um, worsening in severity is what you see with this. So do not get any better. They get worse. All right. This is a sex-linked. This is a sex-linked genetic disorder. What that means is that when you have a sex-linked trait, that means that, for example, in this case, the mother carries the bad gene here. The mother carries this mutated gene, but the mother does not express it. She's just a carrier. And if she has a son, okay, then she will pass that mutated gene on to her son. Okay, so it's sex linked because it's not random, right? It has to be that this is coming from the mother, first of all, and it's only going to go to her son or sons. All right. Now, it says here diagnosed usually between 2 and 10 years of age. All right? Typically, we're going to see this early because, again, like I said, the symptoms you see are going to be very noticeable when the child starts to try to walk. And they're going to demonstrate typically something called the cowering maneuver. I don't think I have that on a slide. You might see that in a clinical scenario on the exam. It's called the cowering maneuver, C-O-W-E-R-I-N-G, cowering. Basically, it's like they're not going to be able to hold themselves up at the trunk because the first muscles to go from this typically are going to be muscles around the hip area, the hip and the legs. So when they try to stand up, when they try to support themselves, they can't stand up straight. They cower. All right, it's like they're kind of like leaning over because they can't hold up their trunk. Right, the muscles are being, the membranes around these muscle cells there are being destroyed. Okay, so cowering maneuver is something you'll see. You'll see them using other objects around them to try to lift themselves up. Right, so maybe they're in a sitting position and maybe they're like next to a wall or a chair or something, and they're going to use that object to try to help themselves get up. So you will see that early. Now, unfortunately, people with this disease, they don't live very long. Okay, they rarely live past 20, and the reason for this is because, well, we have muscle in other areas around vital organs that are going to be affected. And so respiratory is really going to be the system that's most severely affected by this disease and will be the most common cause of death for this person is going to be respiratory failure. Okay, because the musculature around the bronchi, okay, in the lungs fails. So they're just not able to breathe, right? They're not going to be able to inhale and exhale. They won't be able to due to the destruction of these muscle cells around the bronchi. All right, there is no cure for it right now. What else about it? I think I've said a lot of this. Oh, I did say mention the cowering maneuver. Great. All right, so again, remember what dystrophin is. It's basically a protein that's going to link the thin filaments, which are considered part of the cytoskeleton, to the extracellular matrix via or through sarcolemma, right? You will have a fragile sarcolemma, and this also is going to lead to something else I didn't mention. So, because we're having a damage, we're having actual tearing of the sarcolemma, because whenever this person attempts to move, okay, because it's not that they don't have neural stimulation, their neurons are fine. So the neurons are stimulating to the muscle cell, and so automatically the muscle cell is going to try to contract, but this ends up destroying the sarcolemma, okay, because there's no proper structure there. And so because we basically get tears in that plasma membrane, we're having entry of things in there that can do damage. and specifically we're talking about calcium. Okay? So in skeletal muscle, if you have a whole bunch of calcium coming in, it ends up destroying the contractile fibers. It will actually start to sort of calcify these fibers. And this is going to lead to an inflammation response, all right? You're going to have inflammation following this. This is going to be chronic inflammation, and over time, it's just that inflammation by itself, because it continues to stick around, is going to do even further damage to the cellular structures. So again, with all this, you're having a couple of factors here that are contributing to the destruction of muscle tissue. Okay, so over time, this just gets worse and worse and worse and worse, all right? And yeah, there will be a loss of muscle mass. So, this person, is going to try to move because their neurons are telling the muscle cells to move, but it's at the level of the muscle here where, there's going to be damage from this. And so the cowering maneuver, again, is something you're going to see as a symptom. This is really a dead giveaway. If a boy comes in, he's 2 years old or so, and you see this, yeah, it's really a big worry that it's pretty likely that they will have this Duchenne muscular dystrophy. Again, there is no known cure right now. Prednisone or other similar steroids can help a little bit, just by way of their effect on reducing inflammation. There's lots at work here in research right now with trying to figure out how to fix the disease. Of course, stem cells are always promising. What you would do in this case is you would have a population of stem cells. Could be the patient's own stem cells or stem cells coming from another host. And basically what you would do is you would take the proper dystrophin gene, okay, and you would insert that into the genetic makeup of these stem cells and then inject those stem cells into the person. So this is actually showing some promise, so we may see something there. Other attempts have been made. Myoblast transfer, so basically muscle stem cells have been, they've tried to use these, and it just doesn't seem to work very well. And another protein called utrophin, which is kind of like dystrophin, all right, it has this kind of a similar structure. They have been using this in mice, and that's been showing some success. Next disease, we've already really talked about it, so I don't think I need to say a whole lot about it. Myasthenia gravis is an autoimmune disease. We're having antibodies attack the neuromuscular junction. Specifically, we're having antibodies that are directed against our ACH receptors. And so these anti-ACH receptor antibodies are going to destroy these ACH receptors. Okay, so you're getting fewer ACH receptors. Now remember, autoimmune diseases seem to be more common in women. And remember that an autoimmune disease does not show up as a child. Okay, autoimmune disease does not show up in a female usually until sometime between 20 and like 40. All right, 20s, 30s, 40s is when you start to see the onset of an autoimmune disease. So yeah, we have fewer ACH receptors in this case. So our muscles, our muscle fibers will not be as responsive to ACH. So it's not that the person's paralyzed. They're not paralyzed, right? They still have some ACH receptors. It's just that their movements aren't going to be as great, okay? Or it's just sort of like the movement's inhibited. And, you will see weakness, generalized weakness of the limbs. You will have drooping eyelids, difficulty swallowing. You may have double vision. Strabismus is when a person is not able to focus on one point both eyes. So let's say you put a pin in front of their face and you're looking at their eyes and you want to see that those eyes converge onto that one point, that their eyes converge onto that pin. But what you'll notice is that one eye focuses on that pin and the other eye goes in another direction. Okay? Strabismus. Here is just drooping eyelid, right? So basically they're going to have a hard time holding the eyelid up. Treatments, there are a few things, and you can know all these, but I do want you to know that the main treatment here, really, and the best treatment because it has fewer side effects, is going to be this acetylcholinesterase inhibitor, right? It's going to inhibit acetylcholinesterase, which will allow more ACH to remain in that synaptic gap for a longer period of time, to increase the probability that there will be some stimulation of what ACH receptors are still there. Okay, and again, you can look at these. I'm probably not going to ask you about them. They're a little bit more rare, and they can have side effects. So just be familiar with that main treatment there for myasthenia. As far as neuromuscular toxins and paralysis that can affect, right, our muscles, some pesticides actually contain cholinesterase inhibitors. So what this means is that they actually bind to acetylcholinesterase and prevent it from degrading ACH. What this will cause is, in a person who has a normal number of ACH receptors, we're going to have continual activation of these ACH receptors, right? The ACH receptors continue to get activated because you're inhibiting ACHE. So this causes what's known as spastic paralysis. Okay, spastic paralysis. And what this is it's actually a continual contraction of muscle where you don't have a period of relaxation, okay? And that should make sense because if you keep on stimulating that muscle cell, and we talked about this, like in the example of temporal summation, right? Or wave summation, where you can have unfused tetanus or fused tetanus. We're having an increase in the frequency of activation of that muscle cell, right? That's what we're having here. And you have like a sustained contraction. Now, this can lead to suffocation, right? If this goes on, this can actually kill you, okay? Because remember, what we have lining our bronchi and our lungs, right? We have muscle there. So, if you have a continual state of contraction, you could actually suffocate. Tetanus, now tetanus, to mean muscle spasm. Okay, that's what tetanus means, muscle spasm. But there is sort of people will say this is a general term, tetanus, referring to lockjaw. All right, and so basically this is when the muscles of the male will just stay locked up. All right, they stay contracted. All right. Now, so here's the deal: is that there is a neurotransmitter called glycine. Glycine is an inhibitory neurotransmitter, okay? And it's located primarily in the spinal cord. And what glycine helps to do is it helps to sort of put a check or a control on motor neurons. Okay, so glycine helps to actually prevent unwanted muscle contractions. Okay, so glycine sort of helps these motor neurons hone, more specific types of movement or prevent unwanted movement. So the deal is tetanus. Tetanus. What can be a cause of this? Okay. Well, it is a form of spastic paralysis. So you're having a continued contraction. Right. And one cause of this is by a toxin that is made by this bacteria. This bacterium is called Clostridium tetani. Tetani. It's called tetani because it causes tetanus, right? It causes you to experience a fused temporal summation, a fused tetanus. Okay. So what happens is that Clostridium tetani, okay, will actually block glycine. Okay, so your glycine is not able to prevent these unwanted muscle contractions. Okay, so it blocks this inhibitory neurotransmitter, and so then you end up getting this overstimulation by motor neurons that might be doing that. Okay, so we said there's something called spastic paralysis. There's another type of paralysis called flaccid paralysis. Now, this is a situation where the muscles are very limp and just cannot contract. Okay, they cannot contract. Okay. Okay, so an example of this, an example of a toxin that caused this is called curare. Curare is actually a plant poison, right, found in South America. And South American natives used to use this, or still do. Okay, certain tribes in South America still use this stuff, okay, as a poison. They put it in blowguns, okay, they're like little darts, and they blow out of these shoots where these darts contain this curare, okay, and so they can do this if they're hunting or whatever. They will use this. And so what's the effect of it? So curare actually competes with ACH for ACH receptors, and it wins. It wins the competition. So curare is going to basically block the ability of ACH to bind to its receptors. Curare will bind to the ACH receptor, so that means, obviously, the muscle is not going to be able to contract. Okay? Curare does not mimic ACH. It just binds to the ACH receptor, and that prevents ACH from binding, and then you're not going to have muscle contraction. So that's called flaccid paralysis. Botulism. What about botulism? So botulism really is a type of food poisoning. Traditionally, that's what it is. Botulism is caused by the bacterium known as Clostridium botulinum. All right, and Clostridium botulinum makes a toxin, okay, that does have an effect on muscle contraction. Okay, what it's going to do is it blocks the release of ACH from the neuron. This will also lead to flaccid paralysis. Why? Because, well, we don't have ACH binding to the sarcolemma to cause signal muscle contraction. So you're going to have flaccid paralysis. Okay. Now again, this is found in canned foods. All right, if you ever see like a dented can that has a dent in it, or it looks like the can has been damaged, you probably don't want to consume that because Clostridium botulinum might be found there. All right, so you don't want to get that. This relates to Botox, right? You've all heard of Botox. So how does Botox work? Well, Botox has this toxin in it, right? It contains the toxin Clostridium botulinum. And so in small doses, all right, non-lethal or really damaging doses, you can inject these into wrinkle sites. Okay? Inject them into the areas where you see wrinkles on the face, for example. And this will paralyze the muscle in that area. Right now, this is why a person is going to appear to not have wrinkles, because they're not able to move their face. Think about when you laugh and you smile and you're making facial expressions, right? It wrinkles up there, doesn't it? Well, you can't do facial muscle contraction here, right? That's why a person, after they get Botox, you will notice that they are really having a hard time making facial expressions. Of course, too much of this, and it can go systemic, right? You don't want to have too much of this put into your face. It will go to the rest of the body and have effects elsewhere. All right, so the last few slides here are nothing to talk about. These are just these tables I like a lot because they summarize everything you need here, right? So it's giving you skeletal muscle, cardiac muscle, and smooth muscle and comparing them on like where they are, what they look like histologically, so the other structural components. Now, remember, for a lot of this stuff, you don't need to know for cardiac. Okay? You don't need to know mechanism of contraction. You don't need to know that stuff for cardiac muscle because you'll do that next semester. Smooth muscle, a little bit you do want to know about, just whatever we talked about. Okay, so again, you can just look at each of these variables and compare. All right, most important is skeletal muscle for everything, and smooth muscle. And that's it. We just finished chapter 11. It's all I've been talking all day. My mind has been going nonstop all day. So, anyway, I hope you guys have a great night, and we will start chapter 12 very soon. Have a good one.